We go ahead and get started. Thanks to everyone for being here this morning. And we have just two portions of nephrology grand rounds this morning. So Dr. Himmelfarb will speak for the first half hour, and then we'll transition to our last biopsy conference of 2021. So without further ado, Jonathan, please go ahead.
All right. Thanks, Matt, for the opportunity to talk about the work we've been doing in the Kidney Precision Medicine Project over the last 4 years or so. Start with disclosures. And I'm going to start with about 10 years ago, a paper Margaret Hamburg wrote in the New England Journal. This is when she was the commissioner of the FDA, and she used very strong language for an FDA commissioner, saying we are failing as a scientific community to adequately deliver the promise of science to diagnose, treat, prevent, or cure disease, and really put out a call for new methods, assays, standards, and models so that we could speed development of medical products to patients that they are relying on us to provide. So strong language that we need to be doing something differently about 10 years ago from Margaret Hamburg. And that was echoed by Francis Collins, and actually Margaret Hamburg and Francis Collins wrote a few opinion pieces together about how we have to do things differently. Ultimately, it led to a precision medicine initiative, and this paper in the New England Journal, also by a former and the current director of the NIH, sort of launched the precision medicine initiative. And the idea here is to treat every person with a given medical condition as an individual, and not to lump people together into groups, but to say we have to get the right treatment at the right dose to the right patient at the right time. And that means everybody's different. Just because you are in the same category or class of a given disease doesn't mean that you're the same or that we should be treating you the same way. And this is going to require major changes in the way we think about disease process and bring it back to individuals, which is basically what physicians do all the time in clinical practice, or try to do. And from the patient perspective, what patients want to know is, what do I have? What will happen to me? What can I do about it? What does it mean for my family? So this is the call that came from the NIH and from other leaders in American medicine. And this led to the Kidney Precision Medicine Project. And the rationale for why we need a Kidney Precision Medicine Project basically is that the way that we group our major disease entities, AKI and CKD, they're defined pretty imprecisely by the serum creatinine and the urine protein excretion. And we know very well that these two parameters don't cluster patients into distinct disease mechanisms, and that has led to difficulties in our field in creating new therapeutics for a whole host of reasons, including that animal models don't replicate human disease, and we don't understand the heterogeneity of different individuals that have AKI or CKD, which has led to difficulties in prioritizing high-value human targets. And then there's a lack of kidney biopsy tissue to really understand in individuals with CKD or AKI what's going on at the level of the organ system involvement. And this is not new. This has been known for a long time. And Boli was a nephropathologist, and a German nephropathologist who back 30, 40 years ago published a whole series of papers where he showed very convincingly that in that era, it wasn't the eGFR, it was usually the creatinine clearance as a measure of GFR, has nothing to do with what you see on kidney biopsies. And this is just from one of his papers showing perfectly normal glomerulus and an incredibly diseased glomerulus from biopsies of people with identical creatinine clearances. So nowadays we use the eGFR, but you have basically the same problem, which is that you really don't know what's going on in the kidney just by knowing what the eGFR of somebody is. So the rationale for studying chronic kidney disease in KPMP is that 72% of people who end up, if you do a cross-sectional analysis of the dialysis population, roughly 3/4 have disease that's attributed to hypertension or diabetes, disproportionately African American. There are a number of genetic risks in the population. For most people, a standard of care simply doesn't include a kidney biopsy. So we really don't know what's going on or what's driving the disease at a molecular level in that given individual. And of course, now we have SGLT2 inhibitors and also mineralocorticoid antagonists that are coming along, and perhaps GLP-1 agonists. So we are starting to develop therapies, but in the absence of understanding what's going on in the kidney, it's hard to know who will benefit the most from what particular therapy. So the idea of KPMP was that we need research level biopsies, not of the rare diseases, but of the common progressive kidney diseases that are really the public health problem, accounting for most people who develop kidney failure. And for AKI, you could argue that the situation is even worse in that we don't have any therapeutics that are known to be effective. Our definitions are at least as imprecise as the CKD classification system. And there's enormous public health consequences from episodes of AKI. And AKI is still increasing at about 11% per year. Incidence rates in the United States hasn't been going down at all. And we published a paper back, USRDS analysis 12 years ago, I think it was 2009, where we showed that 22% of people who start dialysis have had an episode of AKI in the 6 months preceding the initiation of dialysis. So it's clearly a risk amplifier for end-stage kidney disease, in addition to all the short-term and longer-term morbidity and mortality that's associated with episodes of AKI. So also here too, a great public health need to be able to develop new therapeutics. And it is also not the standard of care or of practice to do clinical biopsies in people with episodes of AKI. So the goals that were stated for the Kidney Precision Medicine Project were to ethically and safely obtain kidney biopsies from individuals with the common causes of CKD that are attributed to diabetes and or hypertension, as well as AKI. Use these biopsies to create a reference kidney atlas in order to find disease subgroups or endophenotypes to stratify patients into different risk categories, and also different who will likely benefit from what types of therapeutics, identify cells, pathways, and targets for novel therapies so that we can individualize treatment and also expand our knowledge, because we don't know very much about what's going on in human CKD and AKI, because it's just not been the standard of care to biopsy the organ that tells us what's really going on. And also that this, the data that will come from this will excite a new generation of investigators to get involved in kidney disease research. And the picture, the photomicrographs, I'm pretty sure Kathy Tuttle gave me these. These are 6 different photomicrographs of people with diabetes and chronic kidney disease, or so-called diabetic kidney disease. And you can see that even just looking at the glomeruli, the pathophysiology is entirely different from individual to individual, and none of that heterogeneity or individuality is captured by the eGFR or the level of proteinuria. So to do this study, we're asking people to take medical risks for altruistic purposes, for the greater good, not necessarily that they're going to benefit themselves from undergoing a kidney biopsy that's being done largely for research purposes. So from the get-go, it was very important to make sure that this was done safely as possible, and under the best ethical considerations. And so there's been a very high level of patient engagement in everything that we do in KPMP. And this paper was just e-published. This is one of our seminal papers in KPMP. Last week, it was e-published in AJKD. First author will be recognizable to most people on this call, our own Kate Butler. And the senior author is also a young investigator who was at the University of Pittsburgh and is now at Penn State, also with an interest in ethics. And the authors include patients, as is typical for our papers, as well as research coordinators, as well as clinicians and ethicists. So it's really diverse authorship. And that's one of the principles of KPMP, is that we really want it to be a resource, particularly for young investigators, to get involved. And so this paper kind of laid out the ethical framework for kidney biopsies and for participation in the KPMP. And because of the nature of this study, from the beginning, we realized it was very important to view patients as equitable partners in the entire operation, the entire consortium. And we have established a community engagement committee that is really central to everything that goes on in KPMP. There's a voting member of the steering committee who is a patient at all times from the community engagement committee. Very unusually for a study of this type, patients generated the first draft of the informed consent form, and really, again, have been involved in everything that we do in the adoption of all the policies there on all our committees, and are very, very active participants as investigators and authors in many of the papers in KPMP. So it's really transforming how we think about a quote unquote patient engagement in research. It's really not patient engagement, it's participants in the study being part of the consortium and essentially part of the investigative team. This is a paper that I think we pay a lot of attention to. Consent is one thing. This is a paper that was published a few years ago in the Journal of Bioethics about establishing trust relationships, especially with diverse populations, for precision medicine research, and certainly for kidney disease. We are clearly, a major goal is to deal with the racial ethnic disparities in outcomes, in incidence and outcomes of both chronic kidney disease and acute kidney injury. And some of these points about discrimination and challenges navigating the healthcare system, influencing trust in research, we take to heart, as well as making sure that data is being used and that participants understand how we're using the data, and also that consent is necessary, but not the whole process of establishing a trust relationship with participants. The consortium this is a very ambitious study, and the consortium is a complex consortium as it's structured. I would venture to say it's probably the most ambitious study that the kidney part of the NIH, the NIDDK, has ever undertaken. There are recruitment sites for AKI and CKD. These are where participants are consented, enrolled, undergo biopsies, and get enrolled essentially in a longitudinal cohort study with biosample collection, tracking outcomes, etc. There are so-called tissue interrogation sites where the biopsy tissue is being analyzed in, as I'll show you, really exquisite depth, using so-called omics kinds of technologies, and I'll describe that in more depth in a minute. There is the central hub, which is a combination of an administrative center, a data coordinating center, and the center that will build the atlas, the so-called data visualization center. And we are it's a federated central hub. The center of the center, so to speak, is here at the UW, but also our partners are at the University of Michigan and Mount Sinai. And the NIDDK set up this study knowing that it was impossible at the time of conception of the study to know what else would be beneficial. So they set up what's called an opportunity pool that allows us, as time goes by, to bring new technologies, new investigators, new cohorts, and new kinds of resources into the consortium on an as-needed basis to enhance the work that the KPMP can do. So at the end of the day, when you look at this complex structure, we now have 200, I think at last count, 295 investigators from 42 or 43 major academic medical centers around the US. So it's a very large, sort of sprawling, ambitious study. And our job is to be at the center of it all and to make the whole thing run effectively. And the gist of the study is it's similar to an observational cohort study with the added fact that people are undergoing a kidney biopsy. So people are enrolled with a given clinical presentation of AKI or CKD meeting inclusion-exclusion criteria for the study. They undergo a biopsy, and as we'll show you, there's not just traditional pathology analyses, but digital and a lot of omics and imaging integration. We'll track clinical outcomes over an extended period of time and use this to accomplish the goals of the study in creating an atlas, understanding disease mechanisms, and identifying basically new therapeutics by really understanding the critical cells, pathways, and targets in these different individuals with these disease syndromes. A key to this is making sure that the data is as pristine as possible and as usable as possible in the long run by a large community. And to do this, we've had to do a lot of work in the background to make sure we all understand the vocabulary that we're using, whether it's to describe different cell types or parts of the kidney. So we have worked very hard to develop a common data vocabulary across KPMP, but also in collaboration with many of the other similar studies that are going on either in the kidney space, other consortial studies, as well as studies like HubMap, which is a trans NIH initiative to create an atlas of the human body, the human biomolecular atlas of the human body, or the human cell atlas, which is also a very ambitious project that's sponsored by the Chan Zuckerberg Institute. And we're all collaborating together to work out in the background what we call ontologies, which is the knowledge basis by which you classify every data element in KPMP. Very vigorous QA, QC, quality assurance, quality control procedures, collecting so-called metadata, which is data about the data, telling you how every sample was handled, processed, everything was measured, etc. So that in the future, anybody can go back and look at the databases that are being developed and the data elements and say, I know exactly how that compares to my data, and I can merge it or meta-analyze it with the data that I'm obtaining as well. So there's a lot of work that goes on in the background to make this data of the highest possible quality. And the atlas that we're envisioning, you know, there's the sort of old Rand McNally atlas, or those of us who are, went to medical school and used the Netter atlas. But think of it as not the Rand McNally atlas, and not even like Google Maps, but closer to Waze, where it's not only how do you get from point A to point B, but what roads are blocked and what's the fastest way to go right now. And so this is really a dynamic living representation of data domains that are linked together spatially to represent the human kidney in these disease states. So it's modeled, if you will, after the pioneering efforts of the Allen Institute here in Seattle, which created the Human Brain Atlas now about 15 years ago. And that's the model upon which all these different atlases that are being developed by the NIH and others right now are modeled after. Very dynamic way of looking at data. And the pathology, there's certainly a traditional pathology read, but also the pathology needs to be computational, it needs to be quantitative, and very, very detailed in KPMP. So we have groups of pathologists. I'll mention Pinaki Sarda, who's at University of Buffalo, Yul Belize, who's at University of Michigan, and Anand Matabushi, who's at Cleveland Clinic. They're leaders in the field of computational kidney histopathology. where you use artificial intelligence and machine learning. And we are basically developing quantitative descriptors and features, hundreds of them that are defined in a kidney biopsy, and then using programs that segment, meaning define the borders for all of these different descriptors and features. You can quantitate every element of the histopathology, and you're kind of seeing on the screen. This doesn't look anything like what we look at when we look at a kidney biopsy, and this is the kidney biopsy of the future, if you will. And then we layer on top of computational approaches and quantitative approaches to understanding the histology, various kinds of so-called omics technologies, whether it's genomics, transcriptomics, proteomics, metabolite profiling, etc. And you can go in, go out on scale. So we look at whole slide images of the entire biopsy, but you can also scale down to look at compartments like the glomerulus, or the tubules, if you will, the vasculature, or get down to the single cell level or even the subcellular level with the data that we're generating. And this is not a new concept. You can see from this paper by Oliver de Voist 7 years ago or so, this idea of multimodal, multidimensional data stacking existed. But what really has been revolutionary in the last 5 or 6 years is the ability to do omics types of analyses on single cells. And that is a game changer in this field. And we're deploying virtually every kind of single cell technology in KPMP that allows you to say, take every single cell, dissociate the tissue. In this case, this is single nuclear RNA-seq that's being done mostly at UCSD and WashU St. Louis for KPMP. And then by looking at the entire transcriptome for each cell and then using these complex clustering analyses, you can define every cell type in the kidney and the cell state based on the transcriptional profile. And that was revolutionary, I would say, about 5 or 6 years ago when we got down to single cell level. But now it's even moving further, and really an innovation in the last year or two is to be able to spatially resolve the transcriptome in situ, inside tissue. And right now this is being done in KPMP through SlideSeq with Anna Greca at the Broad Institute, where you can see here, you can see not just every single cell, but you can look at the neighborhood in which the cell is located, what's around it inside the kidney, and you can look at the transcriptome of that cell. And then investigators here at UW, Ram Alkalesh and Charlie Alpers and Kelly are using nanostring technology, which is a company based here by South Lake Union, probably to resolve even better transcriptional profiles in single cells in formalin-fixed paraffin-embedded tissue. So the capacity nowadays to do amazing things is evolving rapidly, and it really is allowing for the first time an incredibly detailed look at what's going on inside any given piece of tissue, including the kidney biopsy. We also look at protein in the same kind of way. UCSF, Zoltan Lasnik is leading this codex technology, where we're looking again at the single cell level in a variety of proteins expressed inside these kidney biopsies, and using these as anchors really to help define the cell types that are involved. And metabolite profiling is more in its infancy, and it's not at the single cell level of resolution, but it's moving forward. This is being led in KPMP by Kumar Sharma from UT San Antonio, and also Chris Anderton, who's at the Pacific Northwest National Labs. And they're down to about 20 micron resolution of metabolites in the kidney, which is not quite single cell, but they're getting closer and closer to single cell resolution as well. And all these kinds of data now can help us define individual cells and their neighborhoods in the kidney and the cell state, as represented both by transcript and by protein and by metabolite. So it's really, really amazing now to see what can be done with tissue. The challenge, of course, is then to integrate all that data in a meaningful way. And this is sort of, I would say it's underway. It's a little bit in its infancy, but it's moving rapidly in KPMP. So these are some two seminal publications that are in review right now at high impact journals. But here we looked, here's the metabolite profiling looking at metabolic pathways, anaerobic versus aerobic pathways, based on the segment of the nephron and the oxygen tension in that segment of the nephron. We know the kidney is a hypoxic organ, especially in the medulla. And you can see that in the segments of the nephron that are deeper into the kidney, there's much more anaerobic glycolysis compared to more cortical segments of the kidney. And these kinds of analyses have never been done before or possible in human kidneys, as with the technologies that we now have available. And so we can now identify all the cell types in a kidney, and we can spatially locate them and what their neighborhood is, what cells they're near, in both health and disease. And I should mention, in addition to the AKI and CKD biopsies, we're obtaining so-called healthy reference tissue, mostly from a living donor intraoperative biopsies for comparison. So if you know the cell type and you know where it's located, we're also trying to define the cell state. And by this we mean, is it healthy? Is it injured? Is it recovering? What pathways are activated in that process? So these are, again, early analyses, but here from 42, here we combine data from KPMP with data from HubMap and HCA, some of the other major projects that are focusing on developing atlases. And you can see here from 80-odd biopsies, we could define, we analyzed 400,000 cells and 1.2 million cell neighborhoods. So you can see the exquisite level of detail in which you can now interrogate these biopsies. And we're able to define about 100 cell states in health and disease in the kidney in AK, and starting to apply this to essentially redefining AKI and CKD. And this is very early, and I won't dwell on this in detail. I'm also running to the end of the hour. But we're able to now, since we now can identify cell types and look at transcriptional profiles, we can do what's called trajectory analyses, or sometimes called pseudotime analyses, where you look at the cell state based on whether the cell is adapting to injury, whether it's the extent to which it's injured and how it's adapting. Is it repairing? Is it going down inflammatory pathways or regenerative pathways? Is there epithelial mesenchymal transition, etc.? So again, now we're starting to look across healthy tissue, AKI and CKD, and being able to really examine what's going on a cell-by-cell basis, as was never possible before. And there are early suggestions that this will be game-changing. It's still early days for KPMP. It is, we're in year 4. It is being renewed, so there will be at least another 6 years of KPMP, and probably even after that. But we are seeing a lot of convergence between AKI and CKD pathology. So interestingly, because these tissues are processed so well, within 5 minutes of they're time stamped with a research coordinator in the biopsy suite, and from the moment a core is obtained until it's processed and the right fixative is timed and has to be ideally under 5 minutes. So we're really getting pristine quality tissue. And with that, the nephropathologists are looking at CKD biopsies and often saying, I see acute kidney injury. And we're saying, yeah, but the creatinine is at its nadir, and this is chronic kidney disease. So there's a lot of convergence in what we're seeing, not really surprising. injury to the kidney occurs in AKI and CKD, and the molecular pathways are converging too. So these different syndromes probably have far more overlap than we often give credit to. We are adjudicating clinically the cases prior to looking at the biopsy and finding a lot of discordance between clinical adjudication and what the biopsy shows. Again, showing that these large populations that are the people who mostly progress to kidney failure that we don't biopsy, we really have no clue what's actually going on with these people most of the time. And our clinical adjudication without biopsy often changes when we see the biopsy. And although it wasn't anticipated, we are finding that in a fair number of cases, management is being changed by the biopsy findings. So KPMP is designed to be open source data. All the data goes into the public domain, except for data that could identify the participant. It's all obtainable and downloadable from the website. It's designed to be highly collaborative. The data should be used by everybody, and that includes the academic community and also biotech and pharma, so they can make new effective drugs as well. So we're very open to collaboration in multiple different ways. And the data from the 140 or so biopsies is already up in the website in the form of 1,500 data sets that can be downloaded. Another 1,000 data sets should be up by early January. And in the last year, we've had 5,400 downloads from around the world. So people are using the KPMP data. And the goal of this is not just to have data for data miners, people who are going to do these complex bioinformatic computational approaches to data analysis, but to make this available to the broad community, including patients and participants and pathologists and clinicians and different kinds of researchers. A lot of the researchers just want to say, I have my favorite molecule. I want to see if it's upregulated in a certain cell type in AKI, and they can do a lookup of the data and find that out almost instantly. So as time goes by, we're creating tools that go onto the website that allow different kinds of users to use the data, to make it easier for them to put the data in the context in which they would want to see the data that's being made available. So I'll end on time, or 2 minutes late, with the last slide. This is one we use, let's find a breakthrough in kidney health together. So it's still fairly early days for the Kidney Precision Medicine Project, but I do think it has shown a lot of promise that we will learn information that will change the way we think about the common kidney diseases that contribute to kidney failure and to so much morbidity and mortality. At least that's our hope. So I'll stop here, and Matt, you can decide if there's time for questions.
Thanks, Jonathan, for summarizing the, in your words, 1 million man hours that have gone into this over the last few years. And we're obviously going to need an annual update on this, I think, going forward. I think just to be fair to the pathologists, I think we'll move on.